A very good morning to my class, and this is a recording of chapter 16 in BIO 101, the molecular basis of inheritance. So when I am starting to talk about the details of this chapter, just wanted to give you a brief heads up about what we have covered so far. So we have in the past talked about DNA being one of the macromolecules and what are its individual components. Then we talked about the nucleus being the center point of everything in cell biology. Then we were talking about mitosis, wherein we talk about DNA replication and the importance of the exact copy of DNA being supplied to the daughter cells. Then we're talking about meiosis, in which we talk about how DNA replication happens, but how you would have the transfer of only half the number of chromosomes or only one set of chromosomes into the daughter cells that are likely to be formed. And then we talked about Mendelian genetics, where he did not know anything about DNA, but he just called it as particles that are being transferred from the parent to the offspring. And then we talked about chromosomal basis of inheritance, where we talk about if there is a gene in the X chromosome, how is that transferred to the offspring and what is the outcome. We also ended up talking in detail about the foundation that was laid for genetic mapping, or finding out the location of each and every gene in a chromosome. Okay, so putting all of this together, the very important part in this whole story is we kind of sort of know that DNA is the most important unit, and it is the blueprint of life, and so on and so forth. How did we reach this conclusion? Now, we reached this conclusion over a period of time. So in this chapter, I am taking you back to the time where they did not know the importance of DNA. So people were working towards final conclusion, yes, the DNA is the source of information. Okay, and then this is the structure. And how did they reach that this is the structure? How did they conclude towards that? So we're talking about various stories that I have to kind of intersperse this particular chapter with for this process. Okay, now again, a few things that you should know about this chapter is kind of listed out in the first 1 or 2 slides. Okay, so in this particular chapter, we would come across a lot of scientists, and it is ideal for you to know what each and every scientist was responsible for, or what is he known for, or what did he contribute in, why do we need to know about him, or why did we even talk about that person. Okay, so we start with a timeline wherein I'm going to talk about Griffith. Okay, excuse me. So in 1928, okay, let me just go back. In 1928, Griffith was a doctor in the British Army. And during his work, he kind of maintained notes about something curious that happened in his experiments. And that's it. he just had his diary and his notes, and that fell upon the hands of another set of 3 scientists who set about to prove what he did and conclude that it is the DNA that is responsible. So how does this work, or what were the steps that happened in this process? Okay, so for this, the first thing I would like to talk about is everyone has heard of strep throat. Okay, now strep throat is caused actually by an organism called Streptococcus pneumoniae. Okay, now this Streptococcus pneumoniae is a bacteria. Now, Griffith had 2 strains of this. One of them was, when grown on a petri dish, kind of gave him glistening, shiny, mucoid, you can say, colonies. Okay, one strain gave him that, and another strain of Streptococcus pneumoniae gave him dull looking colonies. So that gave him dull colonies. Okay, so when he looked at it under the microscope and stained it with special stains, he kind of knew why. So a little bit about that. So if this is your strep throat, it had like a capsule around it. A capsule. Now, a capsule is a gummy-like substance made up of polymers, sometimes glycoproteins, mostly it is carbohydrates, and this jelly-like substance surrounds the microorganism. And what is the advantage? It can escape the WBCs when it gets into your system. WBCs are white blood corpuscles, which means if it can escape them, it can increase in number, it can multiply and set up a very strong infection. And a person can fall ill. Okay? On the other hand, what happened was the Streptococcus pneumoniae strain that was producing dull colonies did not have a capsule. Okay, now what did he do? He kind of worked with the 2 strains. Just a minute, let me just check if I'm able to get to a picture. Okay, so this is what he worked on, and that is kind of laid out in this picture. So first he had the strep pneumoniae, which is ooey gooey with capsule, shiny colony producing strain. So we call that a smooth strain. In this case he just calls it S. Okay, the ones that produced dull colonies did not have the capsule. He calls that rough strain. Okay, so he took this smooth strain and then he injected it into a mouse, and the mouse died. He took the rough strain, he injected into this mouse, the mouse lived. Now the reason for that, in this case, the bacteria kind of escaped the WBCs because of all the capsule that it had. It was not eaten up, so it increased in number, and then it was able to infect the mouse to a great extent that the mouse died, could not overcome the infection set up by this organism. On the other hand, the rough strain was contained by the immune system of this mouse, and the mouse was healthy and it lived. Okay, then what he did was he took a test tube of the smooth strain, he heated it up. He heated it, and that means he killed the organisms, and then he put it inside the mouse, and the mouse lived. This means there was nothing present alive in it that could escape the WBCs and set up an infection, kind of how it happened in situation 1. So this mouse lived. Then what he did was he put a mixture of dead smooth strain cells from this heat killed sample and live rough strain cells. He put them inside this particular mouse and the mouse died. Then when he looked at the microorganisms, he could see that all of the cells that he extracted from this dead mouse had capsules, which was not the case. The living cells were rough strains and they could even produce shiny colonies on the petri dish. This led him to conclude something from the dead smooth cells got inside the rough cells and gave them the quality or the ability to produce the capsule. Then what happened in 1944, okay, after so many years of Griffith making this particular conclusion, they came across his diary and they worked on it. They came up with a method to prove what Griffith did and to prove that the substance that got inside the rough strain is actually DNA from the smooth strain. So Avery, MacLeod and McCarty announced that the transforming substance which transformed these cells, the rough cells, was DNA. That was the conclusion. But how did they go about doing this? So the first thing they did was they ruled out RNA and protein, kind of like the process of elimination. So they heat inactivated the other two materials in each of the material that they injected into the mouse. For example, in one test tube, if they have DNA, RNA and protein, okay, so DNA, RNA and protein, DNA, RNA and protein. So in this one he kind of inactivated the DNA. In this one he inactivated the RNA. In this one he inactivated the protein. Okay, just a second. Let me just go back. So in each of the tube he inactivated the two elements. So I had DNA, RNA and protein. DNA, RNA and protein. DNA, RNA and protein. So he inactivated two of the elements in each of them, making sure there is only one thing that is active out of the lot. And then he kind of injected them into different samples with the live strain of the rough bacteria and then put it inside the mouse. With this, he noted that only the tube which had active DNA was the one which was able to transform this rough strain into a smooth strain, which meant that DNA is the material that got inside the rough strain bacteria, and somehow this bacteria could read that message and make that jelly-like substance around it, and so it kind of had the quality or acquired the quality or to make the capsule, and so it turned smooth and it turned into a killer basically. So they proved it. It took them 14 years to prove it, and yet many biologists were still skeptical. Okay, then we move on to talk about two scientists. Their names are Hershey and Chase. But before I talk about their experiment, I need to give you a background information about some theory part which would make you understand what their experiment was and how they reached that conclusion. This means I have to talk to you a little bit about viruses. Now, viruses basically are kind of lifeless molecules. They are inactive when they are outside a living system. They are acellular. They don't have a cell. They just have nucleic acid and a protein shell. That's it. Their mode of functioning is they will stay inactive when they are outside a living cell, and once they get inside a living cell of their choice, which is suitable for them, they will set up a system wherein more viruses are being created, and that is how they proliferate. In our case, we need to talk a little bit about each of the kinds of viruses. What is their life cycle a little bit before we move to talk about what Hershey and Chase did. So, viruses, they can be having either DNA or RNA, never both. Okay. And if they have DNA or RNA, there are other options. They can be double-stranded DNA, okay, 2 strands, or single-stranded DNA. They can be double-stranded RNA or single-stranded RNA. Alright, depending on that, they can get classified too. They also can be classified depending on their host nature. So you can have an animal virus, plant virus, and a bacterial virus. A bacterial virus is called a bacteriophage. Okay. Now, the protein shell around the nucleic acid is called the capsid. A capsid is made up of smaller units called capsomeres. So it's like saying I have a brick wall and they are made of bricks. So the analogy I'm like trying to give you is each of the brick that go to make up the entire brick wall is a capsomere. The entire structure is called a capsid. Okay. Now, bacteriophage has certain structure. So it has, this is a simplified structure of a generic bacteriophage. So it can have a head or a capsid. It can have a tail. It has also got a contractile sheath. Now this can behave like a syringe and compress. Okay. It has tail fibers and a base plate with prongs. This is the part that tries to find a spot on the bacterial cell wall in order for it to kind of attach itself, and then make sure that this the contractile sheath that is present is capable of acting like a syringe, wherein there is a plunge where you have the DNA or the nuclear material of the virus being injected inside the bacterial cell. Okay, so here what we have is an electron micrograph picture showing you a bacteria with so many bacteriophages infecting it or trying to infect it. Okay. So I have a video that I have taken from YouTube for you to get a hang of how the bacteriophage infects the bacteria. Okay, so for that, so hopefully that video kind of gave you an idea of how a bacteriophage can seat or find a receptor site on the surface of the bacteria and fix itself. Once it has fixed itself with the prongs on the base plate, it can then organize to kind of like a screw that's turning. It kind of resembled something like that. You would have the entire tail sheath turning through, compressing like a syringe, and making sure in that thirst they can insert the DNA into the bacterial cell. All right, so beyond this, I would like for you to watch the lysogenic video, lytic cycle and lysogenic cycle. What I would like for you to appreciate in that is this video gives you an account of how bacterial virus can take any of two pathways to complete its life cycle. One is called the lytic cycle, the other the lysogenic cycle.
Hey guys, how you doing? This is a video to show you the difference between the lytic and lysogenic cycles of how bacteriophages can reproduce. Just remember that this process, or both these processes, have only to do with bacteriophages, which is viruses that attack bacteria. It doesn't happen in animal viruses like the flu and other things. So basically, to kind of give you a sense of what's going on here, we see that here we have a bacteria. So this is the bacteria right here, and this is the chromosome of the bacteria, and this will be a bacteriophage. This is the virus that attacks the bacteria. So if we kind of see what's going to go on here, this particular virus is going to, as you can see, inject its DNA into the actual bacteria. And so now this bacteria has its own DNA, and it has the DNA from the actual bacteria. And so if we see what happens from this point on, we'll now see that the host DNA has been destroyed. The base of the phage DNA, the genes of the phage DNA directed the host DNA to fall apart. And so now the only DNA that's left in the actual bacteria is the DNA from the virus. And I'm sure you can figure out what's going to happen from this point on. Now, of course, the ribosomes of the bacteria are going to be producing proteins. These proteins are viral proteins, and as you can tell here, the phage DNA is now code for the production of more viruses, and of course now these viruses are going to at some point explode from the cell, and that is basically the lytic cycle. And of course lytic means to lyse, or it means to cut or break open. So this results in the destruction of the bacteria, and that is basically the lytic cycle. So now we're going to go through the lysogenic cycle. So now this is the lysogenic cycle, and this differs in a couple ways, and I'll talk about them after. So the first part of this is the same, that basically a virus is going to attack the bacteria. And so what they didn't show here is that the virus would have landed here, would have incorporated its DNA exactly the same as it did in the lytic cycle, but here's now the difference. In the lysogenic cycle, we see that instead of the DNA from the virus destroying the host DNA, it actually integrates itself into the chromosome of the host DNA. So now the difference is that, there is no destruction yet. So the beautiful thing for the virus is that, well, basically, it's not going to be making a lot of its babies, for lack of a better word, or offspring immediately, but this cell is going to start to divide, and it's going to divide zillions of times. And every time it divides, you can see that little teeny piece of DNA from the phage is going to, it's basically referred to as prophage DNA, is going to be carried from generation to generation. So now you have potentially millions, if not hundreds of millions of bacteria that all have this little piece of viral DNA. So at some point, when this host cell becomes either stressed or exposed to ultraviolet light or some kind of chemicals, a system called phage induction happens. Basically what happens, you'll see here that that little piece of viral DNA pops out, and now basically the cell goes through the lytic cycle. So the second half of the lysogenic cycle is basically the lytic cycle. So what's going to happen? It's going to destroy the host DNA, it's going to start making the proteins, and there we go. So the difference between these systems is that in the lysogenic cycle, the chromosome, or the nuclear DNA material from the virus, incorporates into the actual phage DNA and is carried generation to generation. So the bacteria doesn't make it, or the virus does not make as many offspring as quickly, but it is incorporated its DNA into many, many more cells, and thus giving the ability to potentially make much more offspring over a longer period of time. And so that is basically the difference between the lytic and lysogenic, and I hope you enjoyed it.
Okay, so hopefully that was helpful and informative. Just to let you know, I am using videos from YouTube in order for my class to understand, and I am not profiting from this video that I'm making. It is only purely to help my class understand the material. Okay, so This is a diagrammatic representation of the lytic and the lysogenic cycle. So basically, if we are talking about lytic cycle, the bacteriophage first attaches itself to a certain receptor site on the cell wall of the bacteria, and then it injects its DNA. Once its DNA is in, it kind of takes over the host DNA and destroys it. After that, the whole cell is kind of forced to listen to the directions given by the viral DNA or the bacteriophage DNA. It then directs the cell to make phage material like phage heads, capsids, and then the tail, the base plate, the prongs, the tail fibers, and then there is like an entire factory, like an assembly that takes place. So the heads, the tail, the base plate, the fibers, tail fibers, all of these guys are like assembled to give you a complete phage. And then once that is done, it bursts the host cell and kills it. Now, I had a student in my class who asked me, how do you come to know whether there is lysis that has taken place, or whether it is going to go through lytic cycle, and how do you identify if the phage is so small in size? So I just wanted to give an idea for this. The petri dish will have agar medium and bacteria growing in such great numbers that they can completely form a lawn of growth. Okay, so this whole place is completely covered with bacteria. Okay, once it is covered with bacteria, kind of like this, then you have something known as clearance. Now this is like a very big clearance, I apologize. It's kind of a very small amount of clearance on this place. So you would find a small clearance, and that clearance in this entire lawn of growth is the part where the cells, the bacterial cells, have been killed, and so it is called a plaque. Okay, and these plaques are known to contain a lot of these bacteriophages. Okay, so that is in the lab how you can identify. Well, lytic cycle works like this, and you would have them all assembled lysing the bacteria. So next option that you end up having is, there are some bacteria who do not lyse the cell. They kind of inject their DNA. That DNA integrates itself into the host. Integrates itself into the host DNA and gets multiplied and passed on to the daughter bacterial cells every time that there is a replication and binary fission and bacterial cell division that takes place. So a lot more of the bacteria will end up having this viral DNA. Okay, at certain times, some trigger mechanism like temperature, a sudden temperature increase, or like a temperature shock, salt concentration, some solvents that can be present, these can kind of trigger this viral or bacteriophage DNA to excise out of the host DNA, excise out of the host DNA, and then continue with lytic cycle. So you can have bacteriophages having lytic cycle or lysogenic cycle. Okay, so this is an electron micrograph actually showing you how a bacteriophage can fix itself and inject its DNA into the bacterial cell. Okay, now I would like for you to watch the Hershey Chase experiment, which is a 2-minute video, after I give you an outline. Okay, so think about it like this. The outline works like this. Okay, so there is 2 sets of experiments going on simultaneously. Okay, kind of like if we have like 2 sets of students in the lab. Okay, one set of students runs this and the other set of students runs another. Okay, so first and foremost, this is one line of experiment. First, a bacteria, okay, a host bacteria is grown with supplying radioactive phosphorus in the medium. What will happen is all of the phosphorus sources, which means the bacterial DNA, will be radioactive. Will be radioactive. Why? Because phosphorus is found as a phosphate group in bacterial DNA and it will not be seen in proteins. Okay, and so when I have a bacteriophage infecting this host, this particular host, and lysing it, at the end what it is likely to do is have radioactive DNA. Okay, so this is the first part. Now likewise, on the other hand, okay, another bacterial batch of bacterial host is grown with radioactive sulfur. What is the outcome of that? All of the bacterial proteins are radioactively labeled. And so when I have a bacteriophage infect this bacterial host, okay, what would happen is the bacterial phage will have its entire capsid radioactive. Okay, so now that it has happened, this bacteriophage is now taken and grown in a normal bacteria with normal medium. So this is your normal bacteria, this is the bacteriophage, and this is the radioactive material DNA that it has. And once you give it an initial few seconds or a few minutes to adhere to this surface, you kind of shake it up. What happens is it is capable of sending the phage DNA inside the bacterial cell. And once you shake it up, what happens is you would have the empty shell of the bacteriophage fall off, and the bacterial cell will contain the bacteriophage DNA. This later on, when you centrifuge the sample, the cells being heavy will be in the bottom. The liquid will contain this light bacteriophage head, which is in the liquid, and radioactively will be seen in the cells. In the first case, telling you it is the DNA that has entered the bacterial cell. On the other hand, if this second batch is then treated to a normal bacteria with normal medium, okay, it will try to okay, and then inject its DNA, but remember it has its shell that is radioactive. So the bacterial shell that is radioactive will be, when you shake it up, that is after a few minutes, will be on the outside liquid, and inside the bacterial cell you will have phage DNA which will have no activity. Consequently, when you centrifuge this sample, like I'll put it here, when you centrifuge the cells which settle at the bottom will have no radioactivity, whereas the liquid will have radioactivity because the shell is outside. What is the bottom line of this experiment? It goes to prove that the DNA is the material that went inside the bacteria, took hold of the bacterial cell, and made more bacteriophages. So the DNA is the guy that held the information to do that. So this just proved, one, DNA enters, and DNA is the one that is responsible for making the new bacteria, and so it has the information to do that, and so DNA is the important guy. That's the bottom line. Okay, let's watch this video.
In the Hershey-Chase experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing P32 labeled deoxyribonucleotides. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
Hope that was helpful. Okay, 1 second, let me just go back. So this particular experiment now, I hope, will be very clear and understandable for my students. They are showing you that the radioactive sulfur is pink in color, radioactive phosphorus blue in color. Okay, so the whole experiment is kind of run side by side with the different radioactive material to coat or to tag different macromolecules of the bacterial phage, and to find out what is the outcome, where it finally manages to reach. Okay, so if you notice, in the first batch they have radioactive sulfur, so the radioactivity kind of coated the protein part of the bacteriophage. Okay, on the other hand, they grew it in radioactive phosphorus, so the DNA got the radioactivity. And when they shook it up, the empty protein shells stayed outside, and the phage DNA did not have any radioactivity which was inside the bacterial cell. On the other hand, when they shook the second liquid up, wherein we are talking about the radioactive phosphorus, meaning the radioactivity present in the DNA of the bacteriophage. Alright, so the radioactivity was contained inside the cells of the bacterial host cells. Okay. And so, once again, and so you can see in this particular test tube that because it was the protein shells that were radioactive, they are present in the liquid part of the test tube, and the pellet which contains the bacterial cell and the bacteriophage had no radioactivity in them. On the flip side, if you can see in this one, the liquid contains the phage particles but it is not radioactive. It is the pellet into which the DNA has been injected in the bacterial cell that is radioactive. Okay, so once again. Now, we then have to talk about another scientist. So, Chargaff, okay, Erwin Chargaff had two roles to play and two important points to contribute to the field of science, okay, which helped the future scientists. One, he was a collector. So he collected a lot of specimens and analyzed them. So he analyzed their DNA. What did he find out? When he saw that the nucleotide or the base pair composition was a constant for and specific for each species. Meaning human beings, no matter whose DNA you would extract and see, the base pair composition would be a constant. Whereas a deer DNA that you would extract and check or analyze for its base pair composition, that would be species specific. So basically, the base pair composition is species specific. Okay. The second thing that he found out was the amount of A present in a species is the same amount of T, and the amount of G or guanine is the same as the amount of cytosine. Okay, so meaning if you know the base pair composition of one item, you can know the base pair compositions of the others. Let's try this as an example. Supposing adenine base pair composition is say 35%. So thymine will be 35% if we go by Chargaff's rules. And so what is left is the 30%, which is 15% is for cytosine, and 15% is for guanine. Does it make sense? So this accounts for the base pairs, all of the base pairs that are present in DNA. So you need to know just one and you will know the other three. Okay, so the two things that we concluded, or rather we got as a very helpful contribution from Chargaff, is base pair composition is species specific, is a constant. Second thing that we found out was the amount of adenine is equal to the amount of thymine. The amount of cytosine is equal to the amount of guanine, right? So these are Chargaff's rules. Okay, so people already knew. So now, right now, what we have done, we talked about Griffith and him coming across as something that gets into the cells and gives them a quality. Then we have Avery, MacLeod and McCarty proving it is the DNA that gets in. And then we have Hershey and Chase, again proving and reiterating the fact that it is the DNA that is responsible for information. And then we move to the part where we have Chargaff's rules, where he is talking about base pair composition and that being constant and also being species specific, and also came up with the rule that amount of adenine and thymine are the same in any specific species, and the amount of guanine and cytosine are the same in that same species. Okay, so knowing this, we move to the part now where what some scientists had already worked on. Okay, so what had they done? They already knew that for a DNA or a nucleic acid, 3 components make one monomer. So a base, a sugar, and a phosphate. All 3 come together to give you a nucleotide. So one nucleotide bonding to the next nucleotide, this bonding that exists is a covalent bond. Okay, the challenge was to find the 3D structure that explains all of the quirks and the natures of the nucleic acid that they had known so much about. So we have Morris Wilkins and Rosalind Franklin, who were using the technique called X-ray crystallography. So Rosalind Franklin and Morris Wilkins were students or scientists working in the University of Cambridge. So basically they were extracting DNA and subjecting it to X-ray crystallography techniques and to study the molecular structure. So Franklin had produced a picture of DNA using this technique, and in her raw unpublished notes, she thought that nucleotides were hydrophobic. Okay, so which means they should be on the inside and not exposed. This means that the sugar and the phosphate that are present, which are hydrophilic, they can be on the outside. Okay, so Rosalind Franklin, in her X-ray crystallography plates, these are the results. So she got something like a very constant kind of setup. This kind of resembles a staircase, like a spiral old-fashioned staircase that you're looking at from the top. Okay, so if you're looking at it from the top, you kind of look at it and it kind of resembles this bit. Okay, so everything seems to be very, very evenly spaced out. There is no unevenness in any of these places or abnormal jutting out in any of these places. So she showed her work to James Watson and Francis Crick. When she did, James Watson had already been a person who was familiar with crystallography techniques. He kind of understood that it is like a double strand. Till then, a lot of people had come up with a theory that DNA could be triple stranded. Okay, Linus Pauling, in fact, had put forth a hypothesis that it is 3-stranded. And looking at this picture, which was shown by Rosalind Franklin to him, he kind of knew that it was double-stranded and it was equally spaced out. He also took a sneak peek at Franklin's diary, where she had written about her theory that maybe the bases are on the inside. Okay, with that, he kind of came up with a structure explaining the part wherein he thought that the backbone or the ladder or the sides of the ladder was put together with sugar and phosphate, and the rungs of the ladder were made up of the bases from the opposite strands and the hydrogen bonds that hold them. Okay, and because they were equidistant from each other, he kind of knew that it has to be one purine and one pyrimidine. Okay, and because of Chargaff's rules, he kind of knew that an adenine on the opposite side, it has to bond with a thymine because they are equal in quantity, and a guanine has to bond on the opposite side with the cytosine. And it was also apparent that the structure has to be, in order for it to be stable and in the 3D format, the structure has to be in such a way that one of them is aligned in one direction and the other one is aligned in the exact opposite direction, and that is what gives them the best kind of fit. Okay, so we have Watson and Crick, who built a model out of steel pieces. Okay, and it confirmed everything of the different studies done by different people, and he put forth the structure, saying this is the best way you can explain the structure of DNA. And Francis Crick and James Watson got a Nobel Prize for this. Okay, so the part about the hydrophobic bases on the inside worked out very, very well. Okay, and it was also very clear looking at the X-ray crystallography, which was kind of evenly spaced out, to be very sure that it cannot be two double rings, it cannot be two single rings, it cannot be jutting out and coming back in, and so it was equidistant on both sides, which meant there was one double and one single, meaning one purine and one pyrimidine. So Watson and Crick reasoned that the pairing was very specific and dictated by the base structures. They determined that the adenine paired only with thymine and guanine paired only with cytosine. Okay? And they also kind of figured out that there are two hydrogen bonds between adenine and thymine and three hydrogen bonds between guanine and cytosine. Okay. Okay, so now we come to the part where we are going to talk about the replication of DNA, how the DNA replicates. Again, a lot of scientists were racing to be the ones who were called to be the first to come up with the best explanation for how the DNA replication takes place. Okay, so the basic principle behind that is there is two strands, they are complementary. Now what it meant to be complementary means if one strand had, like for example, think about it like this: if you have a coin and you have the face of some politician and you press it against a mold, you will have the opposite of that here. Meaning, if one strand has A, G, C, T, C, the other strand will have T, C, G, A, G. So it is an exact opposite, or it complements each other in that fashion. So each strand acts as a template for building a new strand in the replication process. In the DNA replication, the parent molecule unwinds and the new daughter strands are built based on the base pairing rules. So a parent molecule kind of separates out the strands get separate, and then you have daughter strands being built, and now in the end you'll have two strands which will have one parental and one daughter strand. There were three different hypotheses put forward in the explanation, or in an effort to explain the replication process. The first one was called the conservative model, wherein the parent strand stays as the parent strand and both of them serve as molds, wherein the parent strand and the daughter strands, there is no connection. I mean, there is a freshly made two strands of daughter strands. That is the theory behind the conservative model, or the parental strand is completely conserved and there is nothing changed, they remain as is. Whereas in the semi-conservative model, the way it works is half of it is conserved as the original parental strand and then a new one is made in complement to it. So it kind of opens up, and now you'll have one parental strand and one daughter strand, one parental strand and one daughter strand, and so on. Okay, so this semi-conservative model. And then dispersive model is like it kind of breaks, and anyone can serve as any part of the template, and there is no hard and fast rule about which part of it is going to be the parent, which part of it is going to be the new. All they can see is it just breaks up and then it is all made up and joins together. But this has room for so much error, and there is great question mark on the efforts of the cell trying to conserve the information and preserve its species nature by having the identical DNA, and the DNA being passed on without any mistakes to the future generations. And that seems to be very doubtful if you follow this method. So conservative method seemed to be the best method, or the best hypothesis. And then we have two scientists, Michelson and Stahl, working towards proving that the DNA replication does indeed take place through the semi-conservative model method. So let me just go back, please. 1 second.
The Michelson and Stahl experiment provides evidence for semi-conservative replication of the DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Messelson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
Okay, hope that was helpful, and now I can talk to you a little more in detail about this. Okay. So the first thing to understand in this is bacteria are first grown in a medium which has got N15. What does N15 mean? N15 means the atomic weight of this nitrogen is higher, right? And so it will be more dense or heavy. So whatever DNA is being made by this bacteria will be heavy. So the idea was, if I first grow it, okay, and then extract the DNA, and then I put it into a gradient of cesium chloride. Cesium chloride, there is a certain way in which, you just have to take my word for it. They use a lab technique wherein they can add the salt of cesium chloride and then centrifuge it. So it kind of, when you make this composition, it kind of separates out into, it's the heaviest to the lightest concentration. The salt kind of forms there. And so in this, if I put the DNA, it will band at a part which is equal to its density. That is what he's trying to say. So in semi-conservative model, what he was trying to say, in the first one, it will kind of band lower than everyone else. Now, if I just have bacteria growing in this, okay, and no N15 at all, N14, that is the normal, it will kind of have, okay, a band which is called the lighter band, because it is not as dense as the dense N15 grown bacterial DNA, right? Now what he says is, if you've continuously been growing it in N15, you then grow it into N14 medium, and immediately the first time, 20 minutes. Why? Because life cycle for this particular bacteria, where one cell becomes two daughter cells, takes approximately 20 minutes, which is E. coli cells that he's talking about. Which means this first time there would have been one generation that has increased, only one cycle that have taken place. At that particular point, what he says is, it is neither very low nor very high. Alright? It is kind of in between. And that is what, and he saw this one band, and which is in between. It's a very important part of the sentence. Okay, one second, let me just go back and kind of put it all together. Okay, once again. Okay, sorry for the wait. Okay, so my point was, if I have in a test tube growing only in N14, the band of DNA will be light in the lighter spot. If it is growing only in N15, the band of DNA will be heavier. But if it has changed from this N15 to an N14, the first batch, it is a very important step. In 20 minutes, if you extract the DNA and centrifuge the sample, you will get, okay, this guy, the N15 guy, meaning, let me just put that here, the N15 guy will have both the strands radioactive. Okay. Okay. Now, after that, he opens this up, and because he is growing in N14, the other one is like this. So, immediately the two daughter cells are going to get this, which means their DNA is neither very dense nor very light, not dense as N15, not light as N14. It is in the middle, and it is only one band he's talking about. After that, what happens is you will keep getting future bands, which will all eventually be more and more and more and more on the lighter side, and there might be just one faint band in the in-between side, and finally this guy will be so faint you will just see one band here, which means all of that radioactivity is kind of gone to faded into insignificance from that one DNA bacterial sample that we started with. My point is, this establishes the fact that the DNA separates out its strands and newer strands are made just to that one template, and so it concludes that semi-conservative replication is the mode of replication. Now, the actual replication process. Okay, so the replication process begins at specific origins of replication, where the two strands of DNA are opening up in a replication bubble. For this, I would like for you to imagine if you had kind of boiled some pasta. This strand is kind of stuck and you're trying to open it in the middle, so you will kind of have one opened up and then the other opened up, so you have like one gap in between. Our replication bubble is kind of sort of like that. Okay, and so bacterial chromosome, it will be just a single stranded circular chromosome. You will just have one replication bubble and it kind of reads each other on either sides. But eukaryotic chromosomes, remember, it has got thousands of origins of replication. Why is that? In order for you to hasten the process of replication. So replication proceeds in both directions till the entire molecule is copied. Okay, so this is what you would see in a bacterial cell, whereas in a eukaryotic cell, you will see multiple pieces of replication or spots of replication. Okay, so now we talk about the different enzymes that are involved in the entire process of replication. First thing that you have is something called as topoisomerase. Now for that, I would like to first talk about what is this DNA. Okay, it is kind of, okay, and then it is kind of wound in over it, and then over it, and then over it. So if I have to unwind the whole thing, if I remove a twist from one side, it kind of twists it more on the other side. So it kind of is likely to break. And to avoid this kind of tension breaking, topoisomerase relieves the tension, so it kind of relieves the gap. Okay, so it kind of opens up the gap. Okay, so what it would do is if one is going over the other and kind of like this, so it would kind of, I do not have a mechanism by which I can erase lightly. I'm going to try. And so this strand kind of passes through out here, and this guy then rejoins at the bottom. So if it was on the top, it kind of comes to the bottom. So my point is, it tries to break or relieve the tension. Okay, topoisomerase. Okay. Now, helicase is another enzyme which untwists. So if I have something like this, so it kind of untwists it and straightens it out. So for that, it is just at the start of a replication fork. Okay. Then you have single-stranded binding proteins which hold a single strand apart. Because remember, it is, first of all, having these bases, it can kind of start bonding with anything else. So it kind of behaves like a sticky tape. You wouldn't want it to behave like a sticky tape and bind anywhere, any which way. And to avoid that, you have single-stranded binding proteins which will bind to single strands of DNA and keep it opened up. Okay. Now, once that is done, then you have an enzyme called primase. This enzyme primase is actually RNA polymerase. And at this particular point, what you have is the DNA is, the tension is relieved, untwisted, single-stranded binding proteins opening it and keeping it open. Then you have primase which has made an RNA primer. Alright? This RNA primer is the first thing that has to be made. It is a short sequence or a segment of RNA primer. Then you have DNA polymerases which then attach to the RNA primer that is being made. They attach the nucleotides to it, meaning they cannot do it individually. They need the RNA primer to first make the primer. So that's a very important part to remember. RNA primer has to be made first. Has to be made. Okay. Now, most DNA polymerases will require this primer and a template strand. And then the elongation takes place about 500 nucleotides per second in bacteria, 50 per second in humans. So in bacteria it is faster, remember. Okay, so this is another picture which kind of tells you that the topoisomerase causes the removal of a swivel. So if they untwist it and it becomes more, if they untwist it here and it becomes more twisted here, it is capable of relieving that tension. Okay, it starts to add the bases, and every time it adds a nucleotide base, it needs the DNA polymerase to help in this process. Now, the elongation takes place in the anti-parallel form. Now, why does this stay anti-parallel? Okay, so I have explained this particular feature many times in different parts of different chapters. One more time. Very important part is your deoxyribose sugar. Okay, so your deoxyribose sugar. So now this is your carbon 1 and it's called 1 prime. This is 2 prime, this is 3 prime, this is 4 prime, and then CH2OH, and this is your 5 prime. Okay, at the 1 prime there is a hydroxyl group, there is a 3 prime hydroxyl group, and there is an hydroxyl group in your 5 prime. Okay, this hydroxyl group is taken over by a phosphate group there, CH2OH. Alright, this hydroxyl group is taken over by a nitrogenous base. So what is left? Only this hydroxyl group is left for future attachments. My point is, in any DNA strand, the attachment of fresh nucleotides take place in the 3' end only. 5' end, nothing can get attached. Okay. So I would like to go through the whole process and show you in a sequence what happens at every step. Then we can watch a video on this also. So the first thing, remember, if I have, this is my, say, DNA strand, and this is my replication bubble. It's kind of opened up a tad bit. I apologize once again. Okay, then this is opened up. Now I'm going to just show this part, wherein first what happens is, at this part you have a primer being formed. Okay, now I need a different color, so I'm going to make this primer. Okay, primer being made on this side. So then what happens is, you have your DNA polymerase adding to it, DNA polymerase adding to it. Now when you look at this, okay, I'm just going to show you this one side of the replication fork. Okay, this kind of gets added without a problem, because as the replication fork opens up, like you unzip a bag and on one side it kind of opens up, you can add without a problem, unhindered. Okay, whereas the opposite strand, so the opposite strand, it is kind of very difficult for it to add, because it has to stumble and go into the 5' to 3' on the opposite side. So this is the 5', once again 5', and this is the 3'. This is my 5' and this is my 3', which means this strand is the 5', this is the 3'. Okay, so if it has to go in the opposite direction, it needs another primer, and then it will kind of form up to this much, because then it comes in contact with this primer that is already there. The DNA polymerase falls off, and then it again needs a primer, and then again it needs to make this, and then it will fall off, and then again it needs a primer, and then again it needs to make this. So it is made in small pieces, whereas the one strand is continuous, no problem. The other strand is made in small pieces. Kind of what I was trying to draw here. So here, if you notice, there is two strands, and then I kind of call them 5' to 3', 5' to 3'. Remember the anti-parallel nature. Okay, so first the primer is being made, 5' to 3' for this side, 5' to 3' for this side on the replication bubble, and then I have a new strand which is being made. On the opposite side, I'll have a new strand being made here. But what about this part of the strand to be copied? It starts with a fresh primer again and goes on to the opposite direction. It has to bump off, fall off, because it comes. It means what? The DNA polymerase has to fall off and then go search of new spot to that it can start to replicate again. So you would have overall one strand which is continuously synthesized called the leading strand, and the other strand that is discontinuous in its formation and it is made in small bits, which is called the lagging strands. And the differences between the leading strand and the lagging strand. Okay, let's just go back and I can put that. Okay, the difference between the leading strand and the lagging strand. Okay, leading, lagging. Leading strand continuously made in bits. Okay, first needs only one primer, need lots of primer. Okay, and then this is faster, this is a little tad bit slower, and these fragments are called Okazaki fragments. Okay, then what else? You will need to kind of dissolve only one primer and then kind of make DNA nucleotides, attach it to that, whereas here you have to dissolve a lot of those primers and then you have to stick it. So you have to stick them all with ligase enzyme. Okay. Also another important difference is in the leading strand, it is made in the direction of the replication fork. Lagging strand is made in the opposite direction of the replication fork, opposite to the replication fork. Okay, so the overview. Okay, you will have small bits of Okazaki fragments on both sides of the daughter strands or both parts of the daughter strands, and then at the end of this you have your DNA. Let me just go back 1 second. DNA ligase. Well, before DNA ligase, DNA polymerase comes into action. It will kind of dissolve this RNA primer, add DNA to it and leave. And then these bits are joined by ligase enzyme. So this structure just kind of shows you what happens at a replication fork. So you have the helicase enzyme at the start of the replication fork. You have the RNA primer being formed by the primase, and then you have your DNA polymerase 3. This is an important part. 1 second, let me just go back. DNA polymerase, the first guy who actually does the extension of the DNA strands, who is able to attach the DNA to the RNA primer, he is called DNA polymerase 3. The at the end of all of the complete copying that has been done, all of the RNA primer is dissolved by DNA polymerase 1, and then in place of that, DNA nucleotides are added to it by this DNA polymerase 1. Once that is done, you still have these gaps. These gaps are then sealed by DNA ligase. Okay. So, the template strand. So first you have the RNA primer, and then the DNA polymerase 3, and then it kind of falls off at every time it hits a primer in the same streak in which it's adding the DNA. And then it will try to find another spot closer to where it can again make an RNA primer, and then kind of reach there, fall off, and then find a spot again. So that is how you would end up having the two strands. So in this table, actually, you have a very good description of all of the enzymes and their function. And the two other enzymes that I need you guys to know is nuclease. Okay. So this particular chart kind of gives you an idea of all the different enzymes that are used and where. Okay, so here you can notice that the DNA ligase is ready to join two strands of DNA that weren't really stuck or joined together. Okay. Then we are talking about the proteins in the DNA replication form a large complex called the DNA replication machine. The DNA replication machine is probably stationary during the replication process, but recent studies support that a model wherein polymerase molecules reel in the parental DNA and extrude out newly made DNA, okay, kind of like a fishing reel that you kind of wind and let the wire go through the wheel. Then there is also something called as proofreading and repairing of DNA. Before this, I would like for you to watch a video on DNA replication. Okay, 1 second.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator.
I apologize. This is a video for protein synthesis. I completely apologize. this is the video I was looking for. So
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
okay, hopefully that was helpful and it ran home the point where you understood the entire process of DNA replication. Okay, right now we move to the part of how proofreading and repairing can happen. So repairing of DNA can be done in two types. One is called mismatch repair of DNA, where you would have repair enzymes which will correct the errors in base pairing. So they kind of look at a certain spot, they kind of highlight it and then replace it with the correct one. So kind of how you do in a computer screen, you would kind of go to a certain spot and highlight it and then replace it with another letter. So kind of like that, mismatch repair can be done. But extensive damage can be repaired by another method called nucleotide excision repair, wherein a nuclease enzyme cuts out and replaces the damaged stretches of DNA. Now, what are all the things that can damage DNA? It can be damaged by chemicals, radioactive emissions, X-rays, UV, and certain molecules in cigarette smoke. Okay, so if you have UV exposure, excuse me, please let me go back. Okay, if you have UV exposure, what happens is in your DNA, if there is a certain spot where there is a lot of T's together, that area is prone to UV damage. What happens is the UV kind of makes the DNA make a mistake in bonding. So T and T will bond and they call a thymine dimer. Okay, when this is formed, there is an abnormality in the structure of the DNA. This is recognized by the nuclease enzyme, which will cut off that portion and then add fresh correct pair of complementary bases by the DNA polymerase 1, which can do this. Remember, DNA polymerase 3 always needs a primer. Okay, so this, what I was talking to you about, is a thymine dimer. Okay, and then DNA ligase would come and seal the issue. So skin cells, on being exposed to the sun's rays or UV rays, can cause formation of thymine dimers, which will cause the helix to lose stability. Okay, lose stability. I have a typo here. Lose stability, and the repair enzymes will keep correcting it. In some, the repair enzymes may be defective. Now, young people, you, me, are constantly having repair enzymes which can function to keep us shipshape. But older you get and more prone genetically you are to get UV exposure, and you are prone not to produce a good set of repair enzymes, those people can be prone to skin cancer called xenoderma pigmentosum. Okay, now we are talking about the next part of the chapter as to why we age. What does that mean? Remember, I was talking to you when I said that we have DNA, okay, 5 prime to 3 prime, 5 prime to 3 prime, right? And when it is making a copy, okay, it kind of makes one copy which is on this side, on this side. Please note that a small bit of this is not copied, Why? Because that is the part which was, once again, I needed to find some color that I can use. Okay, I needed to find this. Let me see if I can do that. So this is the part that was taken up by the primer, right, on both sides. That was an RNA molecule. Remember, RNA nucleotides were there. That is taken off by the DNA polymerase 1. And because it is in the 5 prime end, the body is not, or the enzymes are not able to add, unable to add nucleotides. So that means with every time, every replication, I am getting less of DNA. It's kind of decreasing, that the amount of DNA that is reaching the subsequent daughter cells is a little less. And this means my daughter molecules will get shorter and shorter. Now, does this affect the DNA? There are a few things to remember in this. One, Where all do you think that the cell division takes place and DNA replication takes place? When you are a small cell to the time that you are a baby in the mother's fetus, okay, as the mother's stomach. At this time there is a lot of cell division taking place, okay, and then when you are a baby and to the time that you are an adult, a lot of cell division is taking place. So, which means by the time you have reached this stage, you should not have any DNA. All the DNA should have gotten eroded, right? But that's not the way it works. When you are in the fetus, there is a special enzyme called telomerase. And this enzyme has the ability to add this DNA and bring it back to the normal, original length. Okay? Now, as and when you are growing, are you losing all the important matter? Is the cell losing all the matter that it needs? Not necessarily. Why not necessarily? If you analyze the DNA sequences that we have, in the ends, we have like 6 segments or 6 base pair long segments repeated 200 times, 300 times over. Okay? Or 100 times, depending on where it is, 100 times, 200 times, 300 times over in the edges, the same repeating sequence. These sequences are called telomeres. So this telomerase enzyme has this job of putting back the sequence so we do not get shorter DNA when we are growing from one cell to a baby in our mother's tummy. When we are a growing fetus, this enzyme is present and it kind of puts it back. When we are out and we are growing to be an adult, we do not have this. So our cells have a finite lifetime, which means when these sequences, these useless sequences, but useful in a certain way. Useless because they don't code for anything. Useful because they provide the edges, like the edge of a book or an edge of a paper that you are presenting. Normally, when you would print a paper for an assignment, you wouldn't write at the edges. You would kind of leave the edges free. Think about it like that. And the edge getting cut further. When it gets cut further and it gets into your book, the actual book, that is when you're going to lose information. But till the time, if it is still at the edges of the book, you are not going to lose information. So till the time the cell multiplies 100 times, 200 times over, these telomeric sequences are there to save the day and we are not losing useful information. Beyond that, the cell cannot sustain itself and the cell starts to age and die. This is how we age. So there were some ways in which the cosmetic industry wanted to overcome this and introduce this particular enzyme in their creams, so that when people put this on into their faces, their skin cells would have hopefully the telomeric sequences back and have more youthful look, because they have more lifetime to stay and delay the aging process. But they also found out one flip side, that this telomerase enzyme actually is found in large quantity in cancerous cells, and so we are kind of not really required to kickstart a cancerous process in your skin, right? So telomerase to be used in the cosmetic industry is out, right? So these were your telomeric sequences, okay? TTAGGG, about 100 to 1000 of them are present in the edge of the DNA sequences, which means in any DNA sequence, okay, the edge of the DNA sequence has T, T, A, G, G repeated close to 1,000 times. So every time the cell divides, it loses a little bit, it loses a little bit, it can keep losing till it reaches the normal useful part of the DNA, which is important for information and the normal processes of the cell. Till that part that it reaches there, it is fine and the cell can sustain itself and still continue to live. Once it kind of gets inside, then the actual DNA, useful DNA, is eroded, and this is when the cell dies. Okay, so telomeres do not prevent the shortening of DNA molecules, they postpone the erosion of the genes that are near the ends of the DNA molecule. So this is what is proposed. The shortening of the telomeres is connected to aging. So they also kind of stained the telomeric sequences. So you can see in the chromosomes that it is still present and in a large quantity in each of these chromosomes. So if chromosomes of germ cells became shorter, the essential genes would be missing. So remember, the enzyme telomerase that I was talking about is present, which catalyzes the lengthening of the telomeres in germ cells, in a growing cell, growing baby. Okay, not in a fully grown person. So shortening of telomeres might protect the cells from cancerous growth also by limiting the number of cell division. And there is evidence of telomerase activity in cancerous cells which may allow for cancer cells to persist. So putting telomerase enzyme inside a inside a cream, a beauty cream, face cream, is not advised, is really ill advised. Okay, now coming to the final part of this chapter. In a bacterium, DNA is considered as supercoiled. So it is not only coiled, it is then further folded up. Think of this guy again further folded up against itself. So it is called supercoiled DNA, and this is in the nucleoid region. Chromatin, this is another term. This is so chromatin is a complex of DNA and protein. Histones are proteins that are responsible for the first level of DNA packing in chromatin. Okay, so histones are proteins around which the DNA is wound. So they kind of look, once again, they kind of look like this. So you will have a histone protein and the DNA is wound around this. Then another histone protein, the DNA is wound around it. Histone protein. So it's a complex of a few proteins, but it kind of looks like the way it does here, like a bead. So it's kind of wound around that. So chromatin is a complex of DNA and protein. It is found in the nucleus of eukaryotic DNA. So the binding here, they show, okay, the histones and the way it winds around the histone tail. And the nucleosome will be the DNA and the protein. Further winding or coiling in order to make it prominent, you can see. Okay. So loosely packed chromatin is euchromatin. So I would like for my class to remember something that is easily possible for transcription, euchromatin, because it is loose. Easily in euchromatin, meaning easy because it is loose. Okay. Highly condensed, very tightly packed heterochromatin. Okay. The dense packing of the heterochromatin makes it difficult for the cells to express the genetic information that is coded in these regions. Okay, so in this, just to show how coiling and supercoiling takes place, you have your DNA double helix, you have your histones, you kind of wind, and these are your nucleosomes. You coil first around them and then you supercoil further. So the way it works is you have them here, and then these are your beads, and then this is your bead coiling, and then that going, and that further coiling into one huge tube. This is your supercoiling, finally getting to be your very prominently visible DNA, which is turned into a chromosome. Okay, we finished the chapter on DNA and replication, which is the chapter 16, the molecular basis of one minute. Of inheritance. Okay, thank you, and I hope it was useful.